Ladies and gentlemen, are you ready for a wow? It is with great honor and privilege I introduce to you our 26th annual distinguished speaker, Davis and Davis Professor of Surgery, Pathology, Anatomy, and Cell Biology from Sydney Kimmel. Please join me to give a warm welcome to Dr. Jonathan Brody.
Well, I'm glad, I hope that was recorded because I can just bring that into my annual review, or show it to my wife when she gets upset with me that I didn't change our kid's diaper or something, that I am doing something. But thank you very much. I mean, this has been a wonderful afternoon visiting with some folks, and just the energy in this community here is really palpable. I wonder, I was sort of thinking, hearing these introductions and being out here, because I'm usually in the lab or thinking about grants, I wonder, is this first easy question, want to engage the students, everyone. How many here feel lucky to be here? And I'm not talking about listening to me. I'm talking about being on campus here. And it's okay if you didn't raise your hand, but being here reminds me of my days of being in a small school. And I think then, if someone asked me that question, I actually may not have raised my hand, to be honest with you, to make you realize. But now I appreciate how much this type of education is. And your relationships with your faculty, such as Lakshmi and others around, is amazing. I think it's an incredible experience to engage with these incredibly delightful, intellectual, beautiful people. And so take advantage of that. If I can come here, if you don't learn about pancreatic cancer or DNA or anything else, if there's one thing you take away from the lecture, appreciate what you have right here, right now, because when you get out in the real world, which you've probably heard, it's amazing. If you follow your bliss and all that, but to sit here and think and be amongst intellectuals and do that in a way that is something in the society that usually people don't really people take for granted. Really take advantage of that. So that's my opening line here. So I have a few disclosures about funding sources and other things, and the main thing I wanted to say, which I think was important, I'll get back to, which is what Lakshmi talked about, was I went to college on a music scholarship. So again, I'm trying to relate to the students in the room who might think like, okay, well, this guy's published X amount of publications, he's spent years in school, training, other things. Again, when I was sitting there, I was dreams of being a drummer. And quickly when I learned, coming from a small little town in Maryland and going up to New York with my music scholarship, I realized quickly that trying to make a living as a drummer was going to be a little bit difficult. The competition was tough. And I learned 2 seconds about this, that I learned that I found it really interesting, but unfortunately, I wanted to make a living and I wanted to do something that would have some longevity. Actually really, believe it or not, love playing basketball, even though I'm sort of height challenged here. That wasn't going to happen. That didn't even happen on my JV team. So I learned quickly that there's other ways to find what you love. And I learned that for me, science was learning my scales, if you will, in music, and then trying to be creative with it. And to me, it's the greatest life I know, because there's two things that are involved with it in relation to your college here. It's about education and discovery. And so I get to do education every day, and I get to discover things and study things that no one else does in the world. And to me, that's it's an amazing life, and I'm grateful every day. So I'm going to talk a little bit about cancer. I'm going to go in and out of it. If I get too dense with stuff, I'm going to actually move on and go through the slides. I would love, it seems like everyone right now is really engaged, which is great. I'd really like to keep that engagement. I'd love to make it a dialogue by the end of the lecture. So I'll try to get through some concepts, some ideas that are a little bit different, a little bit out of the box, if you will, about the way we view about cancer and why pancreatic cancer is so bad. So initially, people, I think, I put this slide up here because when people think about cancer and how many people either knew someone, a friend, a loved one, who either was touched, yourself or a family member who was touched by cancer. Okay, so yeah, so you're talking about a disease that, depending on the statistics, 1 in 3 or 1 in 4, we're going to know someone, and you're very young, but we're all old enough that we know either our parents know someone, etc. And it is a disease of age, and I'll get into that a little bit. But I think when we think about cancer, when students think about cancer, we think about it in this sort of lush setting. And one of the things that makes pancreatic cancer really unique, in fact, is that this is the way pancreatic cancer is. It's in a desert. It grows in a desert. It develops in a desert. And why does that make that difficult? I'll let you stop and think about this for a second, because you can imagine that these lush trees, if they get into a harsh setting, they're not going to survive. However, if you move this around the country or around the world into different harsh settings, this is going to survive and grow. And so what pancreatic cancer does as it develops over time, and it takes about 10 or 20 years to develop, it develops in a very harsh setting. And because of that, it can survive a lot of different assaults, including therapies and chemotherapies and targeted agents. So this can grow. And this is really, I think, one of the main reasons why pancreatic cancer is so robust and able to grow in different settings, and what we talk about metastasize, go from a primary site and go live in another neighborhood, if you will, or another organ. So it can go outside the pancreas and live either in the liver or the lung and other places, and that's because it grows up in a very harsh environment. Another reason is for pancreatic cancer, this comes from a review that we wrote. We had an artist come in and they wanted to draw a picture of pancreatic cancer, and me and my colleague, Michael Fishvayan, we explained this to the artist, and the artist drew this picture, which is really brilliant. These are the tumor cells. These are all the different cells that are within a pancreatic cancer. So when Dr. Yo resects a pancreatic tumor, only about 30% of those cells are truly tumor cells. The rest of them are these elements of these microenvironment, these different fibroblasts, immune cells, different stellate cells. These are just names of different cells that are not tumor cells that actually are built into the tumor, and that could be for a whole other 2-hour lecture. I'm going to go on here and continue with my philosophy of what goes on with cancer in general and how we as a research community views it. So there's a little bit of a complicated slide, but bear with me here. This is at the point in which we recognize that there is a tumor. So in this case, a pancreatic tumor. This is years below here on the x-axis, so you can see that minus 20 years is when the initiation of a tumor happens. And this is one of the reasons why pancreatic cancer and many cancers are a disease of people of their 6th and 7th and 8th decades of life, is because this initiation happens so early on. Typically, this starts maybe with some environmental clues, but it happens with a specific mutation in a gene called KRAS. Don't need to know that. But KRAS is a mutation that you might have heard about that really initiates a lot of different cancers, not just pancreatic cancer, but lung cancer, in some instances colon cancer. And you have this initiation event that happens. And I'm going to get into, actually, one of your former distinguished lecturers actually paved the way for this idea. I'm going to talk about that in a minute. But this initiation actually starts the development of cancer. And over time, mutations are gained. And the right amount of mutations are gained that you actually get cancer that gets detected, presents itself in the patient. In pancreatic cancer, it might be jaundice. They might have back pain. They might have a sudden unintended weight loss. They might lose 10, 15 pounds, and the doctor says, well, it's great. Have you been exercising? No. That's a big clue. In fact, we're trying to find ways in which to monitor that on an app so we can detect people earlier. But what happens here is that the cancer is detected, and then we start treating the cancer. And so based on this funnel, you can see that what we do is we start treating the cancer, But then the survival of the fittest, those cells that are able to grow in the desert, can actually live longer, and they go on, and it kills the patient. So this is the current, I would say, overview paradigm that a lot of, whether you're a cancer geneticist, whether you're studying transcription, whether you're studying cell biology, this is an overview of how I think the world views it. So what about the patient? So we have this amazing patient symposium. You're all welcome to come. You can just contact me. It's every November. This past year was November 10th, and this is, some people believe this is the largest group of survivors of pancreatic cancer in a building in the country, and that's all because of Dr. Yo. So we have over 300 people that come, and it's soon to become the second leading cause of cancer-related deaths. The 5-year survival rate on pancreatic cancer, if you get diagnosed, all comers, all stages, is 9%. So just imagine that these patients basically were told, and they, unfortunately nowadays you can just do a Google search, you find out your aunt gets pancreatic cancer, your mom or your dad calls you and says, you're smart, you're at Chestnut Hill, and you just Google it and you say, oh my gosh, they have a 9% chance of living 5 years. And if you're treatable with surgery, that only goes up to about 20 to 25%. So the best chance of a cure is for surgery. Now this is a woman, Margaret Crook, who back in 2007, Dr. Yo, just to give you a side story, Dr. Yo told me about Miss Crook, and I'm allowed to talk about her because she's consented, so there's no HIPAA violation. And she posts on Facebook, believe it or not, her story. But she was diagnosed with pancreatic cancer, but she also had other cancers, and it turns out that she had family members who had cancer. And so Dr. Yo consulted me about it, and I said, let's sequence her tumor. And it turns out she had a specific gene mutation in a gene called BRCA2 that was probably linked to why she had other cancers and why her family members had cancer. And because of that, we put her on a specific cocktail of drug, an agent that specifically targets that, and I'll talk a little bit about it. Anyway, she had a 9% chance to live 5 years. That was in 2007. This is her in 2018. She comes to the symposium every year. When I give my talk, I make her stand up and I embarrass her. It's my favorite part of the year. She had surgery done by Dr. Yo, and she's a lovely woman. So the question is, why can't we get more examples like Margaret Crook? What is really happening out there? So a very talented surgical oncologist, Jordan Winter, and Dr. Avi Nevler, who's a surgeon from Israel, who's now was in my lab and now he's back in my lab. He asked the question to these patients, what do you view, what is a cure? Because we can sit around as academics and sort of say, well, what we think is a cure, is if it's easy for us to say that if a patient was diagnosed, if Alex Trebek lives 5 years, this is a success story. But if you're Alex Trebek, or you're Alex Trebek's wife or child, 5 years really isn't great. Imagine if you were told today that you have 5 years to live. I think you might live your life a little bit differently. You might get up and leave this lecture. But the reality is that this is not a great number, right? And we wanted to ask the question, what do these patients and their caregivers, what do they think a cure is? Let's ask them. They're the ones who are truly affected by it. We're the academics sitting in our ivory towers. So what they said is, you know what? A cure for me is that the person next to me who's the same age as me, my friend, that I live as long as them who's not diagnosed. So we're talking pretty good expectation. And I would say to all of us, we all expect to live as long as our age-related partners. So I think that that's a reasonable request. So we asked the question, we went into a large SEER database, and we asked the question, let's ask people who are diagnosed at either 65 or 70, what is their normal lifespan? past the time that they're, at the time they're 65. Some of them, they live 13, 15 years later. And how is that compared to, let's give the best case scenario, the pancreatic cancer patients that do get surgery put on them. So there's 4 stages in cancer. The 4th stage is the worst. The 1st stage is the best diagnosis. So these are stage 1, by definition, pancreatic cancers. It turns out that only 1 to 2% of these people who have the best treatment option actually meet that criteria. So by definition, by the patients and their families, we are only reaching a cure 1 to 2% of the time. So that, if that's not, to me, more than showing statistics and other things, to me that's very sobering. That's where we're at with pancreatic cancer. And that's why it's one of the most deadly diseases out there. So there's been a lot of work done from the view of what have we done as a community and what do we know? I said in my title that we know so much about this disease. Well, this is a pathology schematic. This is actually done by Jen Parsons, who I was at Hopkins with, and she was the artist there. And this is the way a normal pancreas looks. And what she shows is over those 20 years, when you get the KRAS mutation and the other mutations, this normal pathology becomes dysplastic and transformed into cancer. And you have the right amount of mutations that happen, and this pancreas it becomes pancreatic cancer. And this was all done before 2000, and then after 2000, we had all these fancy next-generation sequence strategies. You hear about all these genomes being sequenced. There's hundreds of pancreatic genomes being sequenced. And she put forth, Jen actually became Jen Parsons to Jim Brumbaugh because she got married, but she put forth this schematic, and it's, asked students, well, what's the difference? Because, you have these mutations here, and everyone says, well, she drew that in blue. The reality is, and the point is that it's the same. It's the same schematic. So if we had the old school sequencing technique versus the new school, we've learned nothing about the genetics in 20 or 30 years. We're just able to do it faster. So we've learned the same sequence over, back in the 90s about the sequence. And I have a little bit of a passion about that because my old mentor at Hopkins actually put this whole thing together. And he did it all basically by hand without this sort of next generation fast sequencing technology. So one of your distinguished speakers, I believe, back in 1999 was Peter Knoll. He might have been at Fox Chase at the time, or Penn, or Wistar. But I noticed when I was actually looking through and learning about the lecture online, number one, I became terrified because the group of people are amazing, and I thought maybe they asked the wrong Brody or something. But one of the things I came up on as one of my idols is Dr. Peter Knoll, who put forth this concept of the clonal selection, which is really part of all my core talks, is this paper that he put forward back in 1976, because it's a really genius theory that goes back to my whole clonal selection theory. And what he sort of put forth is that there's a normal cell that collects the right amount of mutations and moves on and collects. But what's interesting about it is he also put forth this concept that some of these cells actually experiment with the wrong mutations and they die. And there's this whole selection process that has to happen in order for a cancer to arise. And again, we know from recent work that this takes about 20 years to happen for these cells to sort of experiment with the right amount of biology in order to form a tumor. And within that tumor, it's not just the fact that you have one single clone, you have multiple different clones that can actually cause heterogeneity. And he was so forward-thinking because nowadays, I just came back from the American Association of Cancer Research, is they're talking about how this is important for drug resistance. And I wanted to raise my hand at the conference and say, there's this guy who talked about this in 1976. And he wrote here in 1976 in his discussion, hence, each patient's tumor may require individual specific therapy. This is this concept of personalized medicine you might have heard of. And even this may be thwarted by emergence of genetically variant subline resistant to the treatment. So he talks about that there's this resistant clone. I'm putting this forward because I think for a lot of the scholars in the room and myself, we used to have to go to the library and go into the stacks. And I don't know if any of the students know what stacks are, but you'd have to go back and read papers and journals back into the 70s and 80s. And believe it or not, some of these things aren't on Google. And there are pearls here. There are pearls out there that are beyond what's not in Google. So again, one of my little old person's pearls there. So this is a figure from a textbook from the molecular biology of the cell. And this is the reason why, if you look at the incidence of cancer over age, why basically in general, except for childhood malignancies, that cancer is an age-related disease. And again, this goes back to the concept of when I talked about that a normal or a baby pancreas, if you will, to a older pancreas collects these different mutations and goes on and forms a disease. And that's why it's really known that cancer is an age-related disease, because it takes time for these clones to form and actually emerge and present themselves clinically. And this is work done by my good friend Christine Acabuzio, where what she did is some elegant work. What she did was she signed up patients who had pancreatic cancer, and she got biopsies from them and surgical specimens from them when they were actually, at the time, diagnosed. And she basically consented them and said, look, I hope you never see me again. But if this cancer emerges again, I want to come in and biopsy it. Again, I hope you never see me again. But unfortunately, if you die, you're going to have to consent to this. I will drive to wherever the morgue is, and I'm going to remove those cancers. And what she did is, on a series of patients, she called it a rapid autopsy program. She had patients who wanted to dedicate their lives to science, and she actually sequenced the genomes of these tumor cells through the lifetime of that patient. And sometimes it was just 6 months, sometimes it was 2 years. And what she found was that by looking at this, she found that these cancers probably took 15, 20 years to arose. But what she also found is that the right mutations, genetic mutations, genetic events, different than other events that happen, but DNA events that happen in the DNA, the ones that happened initially were the same ones that caused other parts of the disease, and there was no selection for other genetic events. That being said, that all the genetic events that happened when the patient presented with its disease initially was the only events that were necessary. And so that there was something else than genetics that caused these tumors to recur and actually kill the patient. So we still believe, because of that knowledge, it's important to know what the genetic landscape is here. And I took part in a program that was across the country. It was over 48 states, where it was called Know Your Tumor. And it was set up by Pancreatic Cancer Action Network. And what we did was no matter wherever the patient was, whatever lifestyle they had, whatever insurance policy they had, it didn't matter. If they were diagnosed with pancreatic cancer in South Dakota, Atlanta, Oregon, Idaho, we told them we would sequence their tumor. And what we were able to consent 1,200 patients to this. And what we found is that out of those 1,200 patients, we found that 27% of these patients actually had mutations that were actionable. And what that means is that there were drugs out there, FDA approved, that could actually target these mutations. And so this was pretty inspiring, because these are patients who didn't have access to clinical trials. They barely had access to chemotherapy. They would see physicians that never had seen a pancreatic cancer before. Again, Lakshmi nicely brought up, but 55,000 people who are being diagnosed for pancreatic cancer each year, even though it's becoming the second leading cause of cancer-related deaths in this country, is not a lot of people. If you tell the pharmaceutical industry that you're working on something that's affecting 55,000 people in the United States, they would hang the phone up on me, right? Because this isn't a big enough pool of people to care about. However, of those 55,000 people, if you tell a quarter of them, you know what, we have a specific drug that's going to target your cancer, I think those people and their families would be pretty happy. So we found this to be pretty interesting. We published this in Clinical Cancer Research this past year if you want to look at those findings. And one of those things that are actionable, that means things that it can target, is this same thing that saved Mrs. Crook, is this BRCA2 status. And this was work that I was involved with at Hopkins. And there's this really interesting idea called synthetic lethality. And what synthetic lethality is this concept of, let's take one of the chairs you're sitting on. Perfect example of what happens in a tumor. If you take the chair you're sitting on and you remove one of the legs, you'll probably be able to manage to sit on it, right? And so that's what happens in tumors that have BRCA2. Removing that leg probably sets the stage for them to be mutated and have cancer. What synthetic lethality is if you remove another leg, it goes down. There's nothing you can do. It'd be hard to balance yourself on it. And so that's the concept of synthetic lethality. And this is what we found happens in these tumors is this is a tumor growing in a mouse, and you can see it grows very nicely in the mouse. Again, these mice are just bred for these experiments. This doesn't have the BRCA2 mutation. So what happens is when we treat with a specific agent against that, you can see that the tumor regresses. If we actually correct, whether it's through something you might have heard of, CRISPR technology or overexpression with the virus, this gene, the tumor becomes resistant to the therapy, thus proving to us that it's that one gene in 35,000 or so genes that actually make it this susceptible to the specific DNA damaging therapy. And of course, we found that other drugs didn't work as much. Another example that I was part of a study that was really exciting is we had a patient who came in who was diagnosed as 63-year-old female with a family history of cancer. Because of that family history, we went in and we sequenced her tumor. We found that she had a BRCA2 mutation. We put her on a specific therapy, in this case something called PARP inhibitors, and basically this woman had a complete response here. And that complete response was amazing. However, after a year of really responding, when she shouldn't have been alive after a month or two, she recurred with the cancer. And so 25 months later, when she recurred with the cancer, we removed the tumor and we sequenced the tumor. And what we found, which was pretty remarkable, is that tumor found a way to revert its mutation back to a wild type. So the mutation that actually probably caused the tumor, got it to be sensitive to the therapy, ended up reverting back into, it was a truncated mutation for the real molecular aficionados out there, and what it did was it put it back in frame to read it all the way to the stop codon. So what we found in this woman's tumor is that the tumor said, you know what, enough of this therapy, I'm going to change my DNA, I'm going to re-mutate back into a normal form so you can't do anything to me. And that's what caused the tumor. Really remarkable stuff, because from an intellectual standpoint, it showed that we are truly targeting the lesion. From a sobering standpoint, we found that we needed a way to now find a way to stay on top of it and play chess against the tumor. And I'm currently writing a grant right now with Israel to try to find ways in which to stay on top of this. So as a side note, I thought I'd bring this up, and Lakshmi brought it up, that how many people in the room are either taking or took molecular biology? Part of them, so there's a number. How many people have run DNA agarose gels? So how many people use TBE or TAE, tris boric acid? Okay, so that's bad to raise your hand there, because we developed this new buffer called lithium boric acid that you should look into. And the reason I brought that up is because this is actually me as a postdoc with a little, at least I thought I had more hair, so I had bigger sideburns. And this is with my mentor, Scott Kern. And what we found was there was a decimal point error in a protocol, and because of that, we found empirically that there is a better way in which to run a DNA gel, that you could actually do it in 10 minutes instead of waiting an hour or two. So something to look for, and then we ended up, based on this, publishing a number of these papers. If you're really interested in looking those up, you can look them up, and we've been cited a number of times and have had patents on this as well. And so we can use that to really have this value again of potentially molecular profiling and looking for these actionable mutations. And so what are we doing with that information? Well, we're setting up a clinical trial in which we want to really over those 48 states, or really over everyone, we want to capture what patient has that specific therapy out there and set up these little clinical trials and ask the question, will patients do really better if they get this sort of targeted personal treatment going on? Other ways that we're looking at this in the laboratory is that one of these genes that's mutated is called P16. I'm not going to get into the signaling pathway there, but what that means is that you could use specific FDA-approved drugs such as CDK4 inhibitors. This is not published. We have this under review. And what we find here is that in a P16 deficient tumor in vivo, if we treat specifically with this inhibitor, we can slow tumor growth down compared to the controls. So that's where we're at as far as precision therapy is concerned. I'm going to be mindful of the time. I'm not going to go over, if I keep going too much, I'll get to the end of it so we can get to some provocative questions and discussion. But really, this is this concept of precision therapy and precision oncology that I presented to you. It's profiling the patient's tumor and trying to match that tumor. And how are we going to make the difference here? So what are the obvious assumptions that are out there if you're a cancer researcher or a pancreatic cancer researcher? Well, you need to understand the microenvironment. I talked about that with the desert. It grows in a desert, so we need to pay attention to the desert, even if we know the actionable mutations. Even though we do have these targeted therapies, we need to develop better targeted therapies. We need early detection, so we need to move that needle backwards on that 20 years so we can actually get patients to surgery before the patients become metastatic. The neoadjuvant stuff I'll save for Dr. Yo's next talk, and what that means is that it's out there that maybe you should give chemotherapy before you give surgery. And we need better models, and I'll talk a little bit about that. How do we study these tumors in the laboratory? And the non-obvious assumptions that I'll end with today is we need to look beyond the genomics. So one of the points I wanted to get across today is that the reason why cancer researchers, I think there's been a little bit of a halt in our knowledge is we've relied so much on the genetics. So I grew up a cancer geneticist being at Hopkins, and it turns out that all that sequencing, we've learned the same thing over and over again. And we need to look beyond the DNA because we know the DNA is not important for selecting for when cells leave the primary tumor and go off into other sites and kill the patient. And we need to think about rapid tumor evolution. How do we rethink and define our targets? So yes, those tumors are growing in a desert environment, but then we start giving chemotherapy and a storm starts coming through and everything. How does that, that sandstorm, how do those cells protect themselves in that environment? So we have a number of different things going on that we're focusing on to look at this in different ongoing work with different institutions that each one of these I could give a separate talk on, and I'll touch on a little bit. But one of the points I put this slide in here is one of the things I'm really excited about, and I'm actually going to Portland tomorrow to talk to my friends at Oregon Health Sciences University, is this concept of patient-derived models of cancer. So I brought up mouse models, I brought up cell lines. Some of you in the audience who are interested in cancer research or just how do we study cancer, how do people study cancer, what we typically do, what it's been based on is that we try to get tissue from patients, but we also have mouse models. And one of the things that we try to do is we have things called cell lines. And so these cell lines are cells that are grown from a patient's tumor, and we grow them in flasks, and we do experiments in flasks. And some people find that very artificial. And so what people typically say is there's different models, and you literally lie with the models that you can play with to do experiments in. Because if you take cancer cells in a petri dish, I can kill those cancer cells with enough Coca-Cola. We know if we give Coca-Cola to patients, it's not going to do anything. So we need to find the right model. And so what we're doing, we just got a really nice score with Oregon Health Sciences University, a $2.5-ish million grant, to ask the question, what is the best model out there? And so what we're doing is we're taking the patient's tumor and we're making these different models. And the models out there is, one's called an orthotopic model. And what the orthotopic mouse model is, we take the human cancer cells and we inject them into the mouse pancreas. And in fact, they actually grow out as if they do in a human. It's kind of interesting. Another one is this thing you might have heard of called 3D organoids. And these are in 3D, so they're a little more than 2D. They actually grow in different layers in a matrix, and it looks like this, so you can almost look like a pancreas organ on a dish. And another one's called conditionally reprogrammed cancer cells. And we're asking the question from clinical trials, how much do these models actually replicate what happens in the patient, simply put, and can we learn something from it? So again, like I talked about, this is really what the field's been focused on. And one of the reasons why it's been focused on the central dogma of cancer is this DNA is because if someone tells you, if you want to be a cancer geneticist, listen up, this is it. 100 pancreatic tumors, 95 of them have a mutation in KRAS. 100 normal pancreas, 0 have a mutation in pancreas. That's a oncogene. You're a cancer geneticist, you passed the course, my course. That's called an oncogene. If you have 2 alleles that are lost to the gene, that's a higher frequency than in the normal, that's a tumor suppressor gene. That's cancer geneticists. No one's going to argue with you if you find those 95 out of 100. However, when you start studying biology, you start studying trends, and you start studying things that are a little bit more difficult, it's harder to prove to people. So we as scientists like to label things, and because we can label these genetic factors, a lot of scientists study these things. This is a very crowded field. And so I think this is just as important and rapid, and so this is what my lab study. My lab likes to study when the RNA is made, how is it made into a protein, and this is, again, for those not totally focused on biology, DNA is made into RNA into protein, and this makes up the human being. This is what we study, post-transcriptional gene regulation, post-RNA. And I think this is important because I think even though a lot of people have studied this, it is really what happens here rapidly, not over decades, of how these cancer cells can adapt to these environments. They're not relying on genetics all the time. They're relying on other things. And so just like in life, being a little philosophical here and thinking about either whether you're a psychology major or other things, there are intrinsic and extrinsic stressors, right? We all know that the test you have tomorrow is a stressor. Why am I sitting here listening to Brody talk? Versus your own anxiety of how much pressure am I putting on myself. I really know this stuff, but I still don't want to be sitting here listening to Brody talk. And so those come together, and there's a selective pressure that happens. And I would say to you as an analogy that cancer cells have this same selective pressure. And it's the survival of the fittest who end up graduating and moving on and being successful at that time, who can go on and become clonal, and then those cells actually are ready to take on the drugs. We can kill 90 to 95% of these cancer cells, but it's the 5% that are really good at taking care of these intrinsic and extrinsic pressures that allow them to move forward. And so this is the protein that we study. It's called HUR, or HER, and what it does is it basically responds very rapidly to stress. And the way it does that is it moves from the nucleus to the cytoplasm, and it takes with it and amplifies a pro-survival network. So it's basically in a car crash, it's the airbag that allows the cancer cell to live. And so here's another way of looking at it. We've proven in a number of papers that pancreatic cancer-associated stressors basically push a button on the pancreatic cancer cell, move this protein from the nucleus to the cytoplasm, and amplify this whole genome network of transcripts to survive under these different environments. So it's important for evading the immune system, causing metastasis, chemo resistance, a number of things. And I'm going to just give you a couple of examples of things that really show this dramatically. So one of the things that we know that pancreatic cancer cells live in is a very hypoxic environment. So normoxia is just normal oxygen, about 5% oxygen, in which we need. But however, when you look at the tumor cell, the pancreatic cell, it lives in 1.1% oxygen. This is another example of that desert setting that pancreatic cancer cells live in. So if you look in the normal normoxia conditions, this is another play on the models we use. This is a pancreatic cancer cell. If we give that pancreatic cancer cell a known chemotherapeutic, oxaliplatin, which is given in the clinic every day, this green marker here, and the blue is just to outline the cell, is a marker for DNA damage. It means that the cell's about to die. There's a lot of stress here, okay? If we put that cancer cell into a condition that's similar to what happens inside a patient, there is no DNA damage here, none whatsoever. So we found that this HUR protein was critical for that, because if we silence this HUR protein, and again for the molecular aficionados, we use an siRNA molecule, so an oligo targeted against HUR transcription, we can see that we turn on the cell's ability to die. And that's a very dramatic view. Same thing happens in glucose. So we know that pancreatic cancer cells live in low levels of glucose, okay? And we find that, again, if you give a common chemotherapeutic under normal conditions that don't reside what happens in the patient, we can kill the cells, much like the Coca-Cola on the cells. However, we put them into a low glucose setting, these cells don't die. This is what happens in the patient. If we, again, silence this one gene, we can get those cells to die. Very dramatic. I'm going to go through some of this stuff rather fast because in the interest of time. So the question could be is how can we use this technology and this understanding of this HUR protein? So one of the examples is we could actually target one of these downstream events of HUR, but another example is we could potentially target HUR. So one of the things is we need to look deeper. How does HUR regulate the genome? So when we talk about, because I think there's some people, some students, the molecular aficionados out there, when we talk about genes, when we talk about these genes, what part of the genome are we talking about that's being made from RNA into protein? So how many people think coding region? So the coding region of the gene, right? So, right, coding region of the gene makes the amino acids, right? So this is very simple. How much of the coding region, so if this was the whole genome right here, how much of this whole top thing would be the coding region of our genome, of your genome? Very small amount, thank you. So only this small amount. What's going on the rest of the genome here, the non-coding region? So the way HUR regulates these transcripts is in the non-coding region. So when you think that these people went and sequenced pancreatic genomes, do you think they sequenced this part or this part? They just sequenced this part. Because why sequence this part? We don't understand it, right? So I don't want to sequence something I don't know, and I can't get funding for that, so why would I do that? So it turns out that HUR regulates these genes in this part of the genome. And what's exciting is we need to dive a little deeper. So my father's a psychiatrist, so I always put this joke up here for him. But the reality is what we found is that HUR regulates one of these key genes called WEE1, and WEE1 was actually discovered by Paul Nurse, who was a Nobel Prize winner. And he called it WEE1 because he discovered, I think, up near Scotland, and it was like he mutated either a bacteria or a phage, and it was WEE, so he called it WEE, and that's why it's called WEE1. It's an important cell cycle regulator. And to cut to the story, we found that HUR regulates this non-coding region of WEE1. And it turns out that in that non-coding region, when my technician gave me the sequence, he kept saying, I can't clone it because I keep getting this weird variant within the non-coding region. It doesn't make sense. It doesn't match up with what's supposed to be out there. And I started saying, well, maybe this is interesting. Let's sequence other pancreatic cancers. Let's look at this. And sure enough, what we had found, and we haven't published this, we look at the 1,000 gene populations, the European population, our own population, to really summarize a lot of stories. What we found is in that this region of the genome, again, this region of the genome, we found that there is a high prevalence of this sequence in patients who either have family members who have certain types of cancer. And this would never be discovered if we just focused on this in the box, if you will, region of the coding region. So the point here is, through the biology, we found a sequence that was really interesting and actually could be an early detection marker for not just pancreatic cancer, but colon cancer. So stay tuned for that one. And I'm just going to end with, really this question of HUR, because if you can target the airbag sequence, if you will, if you could target the airbag of a pancreatic cancer cell, so when they get the treatment and only 5% of the cells can live, you could actually maybe do some damage against these cells. This is just to show the way we look at things. This is called immunohistochemistry. These are basically 100 pancreatic cancers. And what we found here is that virtually 80% of them overexpress this HUR protein when we resect them from patients, so it's highly abundant. We also found, Chris Schultz, who's a talented graduate student, and Sammy Brown, that during the sequence I talked about, this normal to the progression model, as you go through time, whether it's the mouse or the human, you get an increase in HUR expression over time. And if you actually cross in a mouse a KRAS mutation with an overexpression of HUR, you get more tumors. And Sammy found this, and we published this this past year. Now, this is a real fascinating thing to sort of, I'm coming to the end here, to think about. So some of you heard about CRISPR technology. This is a technology, basically the immune system of a bacteria that can actually target and knock out a specific gene. So what we did is we took a number of tumors and we knocked out HUR. And we knocked out HUR and we grew them in cells that were grown in normoxia, normal glucose and everything. And basically those cells grew fine. Can anyone tell me why those cells grew fine if this is such an important factor? So the reason they grew fine is because they were in this normoxic high glucose condition. But what happens if we put them into a hypoxic condition and we put them into mice? Well, it turns out that they don't grow in mice when you remove the gene. And this is the cells growing here on the line. Why, if you just knock out one of the genes, you get a little tumor growth. Keep both of the genes, you get a lot of tumor growth. And so we did this in a number of cells grown from different patients, giving us Shruti Lal, giving us the proof of principle work. She did all this hard work to show us that when you silence and completely remove HUR, these cells will not grow in an animal. So what we quickly did is we screened for a number of agents that might target HUR. And what Chris Schultz here, who's a graduate student in my lab, what we found was a drug called Providium pomoate, which was an FDA-approved drug in the 70s that actually targeted pinworms. And it turns out that this drug specifically not only targets pinworms, but it targets HUR. And so much so that this is still available in Finland. And he was telling me about it one day, and just to show you how inspiring Chris is, because one day I went by to see him, and he has his Cheerios by his, this is his desk. And here's his poster and everything, and here's his tie for occasionally when he needs. But he had this little yellow boxer, and I realized he bought Providium online from Finland. And he started treating cells, and he came to me and said, Dr. Brody, this stuff is amazing. In nanomolar concentrations, we can kill pancreatic cancer cells. This is 10 to 15 times more potent than what's being used in the clinic. And so sure enough, we use this HUR inhibitor, and we show that they're completely sensitive nanomolar ratios. We show that it actually targets HUR. We showed that the mechanism works. This is called a ribonucleoprotein immunoprecipitation. When you add in the Providium pamoate, you block HUR's ability to act as that safety valve, if you will, where it acts here and increases the survival transcript. We showed that it got into cells. We went in vivo and we just showed that single doses as a monotherapy, that it actually killed tumor. This is growth on the size. It actually killed pancreatic tumors compared to the ones that didn't. And this is actually the control that's being used in the clinic. So you can see how well that's working. And because of this, we're setting up what we call a very innovative window of opportunity trial. So what we're doing is 2 to 3 weeks before Dr. Yo takes a patient into surgery, we're asking them to take some pinworm medication, and then we're going to look at their tumor at the time of surgery and see if that pinworm medication that has a fluorescent label on it gets to the tumor. And then we're going to follow those patients out and actually see if they have the ability to respond to this. And why is this important? It's important not just because we might have found a real drug and a great target, but it's important because if we here all collectively used our intellect and came up with a great target and a great idea for pancreatic cancer, it would take us roughly 15 years and $1 billion to get that idea into clinic. If you think about all the regulations, all the legwork that needs to be done. So finding something like an FDA-approved drug that's for something for pinworms that might just get the worst toxicity is get someone some diarrhea is, I think, incredibly important. Other ways we're targeting this, we're working with a company that's not too far from here called Genosphere, where we're using a nanoparticle. It's a nanocarrier. It's 3 DNAs mixed together like one of those jigsaw puzzles, and we can put different molecules like the Providium or an SIHUR tethered to it and deliver it to cells. And we can do this in an orthotopic model that we talked about, and I'll just show you the example of what we've done. So Grace McCarthy's injected pancreatic cancer cells into the mice, and she shows that by injecting this nanoparticle, she can target these tumor cells by just bioimaging these mice outside while they're still alive. And then we can show that those tumor cells actually can go to specific organs and we can treat them, and that's what we plan to do. So that's our ability to just do one single dose of an SIHUR and keep these mice living longer. And we hope to bring this nanocarrier treatment to the clinic in the next couple of years. So my take-home points is you need to look beyond the genomics and the transcriptomics. Obviously, I believe in the genomics. That was the first part of my talk. We need to think about how rapid tumor evolution and how to rethink and redefine our targets. Just looking at the genetics and how they form over 20 years is enough. We have to think about, as a hospital, as doctors, when we treat these cancer cells, we're putting a selective pressure on them. We need to go beyond the coding region. I think this might be important. So I don't call precision medicine personalized therapy. I like to call it present focus therapy. We need to think about how these things become addicted to this type of post-transcriptional rapid gene regulation. This mechanism happens within 2 to 4 hours after treatment in the laboratory. And understanding these mechanisms will allow us to find new targets such as HUR that I talked about. And so we have these different, based on all this technology, we have these different things that are about ready for phase 1 trials at Thomas Jefferson, and we're very excited about that. So, as mentioned from Lakshmi, my partner, if you will, my producer, my patron, one of my idols is Dr. Yo here. This is the lab. Samantha Brown is a PhD student, very talented. Alex Haber, Aditi and Lee are both working on the DNA damage work. Grace and Chris, I talked about their work, and Tina, they're all trying to target HUR as well. Joe's been with me. He lost his wife to pancreatic cancer. He's been with me from day one, if you will, and he stayed with me. And Monique is a lab manager. And then Sammy's named my son Ethan the lab mascot, if you will. So we'll have him gloved up and in the lab, and I think next year. So what needs to happen to make a difference, just to end with here, and thank you for your time and your attention to listening to me. I think funding, and more funding. I was talking earlier that I want people to realize this is the greatest life to discover and work with smart people and move forward, but still, out of 10 grants that go to the NIH, only 1 gets funded. That's for every 10 ideas, only 1 gets funded. And I think we just need to keep beating the drum on this, if you will, to sort of have the government put more funding back in, as well as have grassroots organizations to support it. Awareness, I think Lakshmi inviting 2 people who study pancreatic cancer for this, it's admirable. I love that. Thank you. I think this is the type of effort that people don't focus on, so I think it's great. And then finally, I just want to say, smart young people like yourselves in the audience who want to take on this battle with tremendous dedication and grit, I think that's going to make the difference. So hopefully I've inspired you to think about maybe going to science, and if it's not science, think out of the box and do what you love. Thank you. Thank you. I think we have a little time for questions. Yes?
So you don't need CRISPR to knock out HER, the pinworm medicine, correct?
Correct. It's a small molecule.
What other cells would produce HER?
So great question. So it's ubiquitously expressed. It means it's expressed everywhere. However, it's overexpressed in cancer cells, and that's for another topic. We think we know why.
Temporarily knock out all the HER.
Yeah.
And not make the person sicker in another way.
So 2 things. So one is we think in mice and other, we find that it's just, there's higher levels in cancer. The other thing is with the reason we're doing the nanoparticle, the dendrimer, I didn't get into it, is we have a targeting moiety that will specifically deliver the drug and the SIHR specifically to the cancer cells. But we think the therapeutic window there is that cancer cells overproduce the HER protein. That's why we're interested in the first place, to survive. And other cells don't need it. Other cells need it initially during embryogenesis. It's actually embryonic lethal. That means that if you knock it out in mice, those mice can't live past early stages of embryogenesis. Yeah, please.
Have people in 25 words or less describe how was it Chris figured out that pinworm medicine knocked out HER? How?
Okay. So 2 ways really we got at that. One is we did a screen, so it's called a drug screen, and typically nowadays you can get FDA approved drugs and set up a platform or an assay to sort of say, okay, I'm going to screen 1 million compounds and find out a way in which one actually targets my gene. And so that's a complicated molecular assay, but actually not that complicated. So you can look at a plate and sort of say, oh, there's a yellow dot there instead of a green. That's probably targeting it. And then you validate it. The other way is he actually just did, again, going back to the stacks, a literature search. And he found that someone somewhere had found that it also targets HUR in other cancers, didn't really explore it. And he also, basically found that people had reported in very, not really recognizable journals that this thing might have an anti-cancer effect, and they never followed up on it. So just through old literature searches and our compound screening. Others? In the back.
I'm going to ask the non-molecular biology question.
Please.
In the very beginning, you had commented that you have a higher survival rate if you have surgery versus non-surgery. Who's getting surgery and who's not getting surgery? Where's that decision process?
Okay, so that's a great question because the easy answer, the scientific question is By definition, if you present metastatic, that means that the cancer is outside your pancreas. The literature shows that you should not get surgery. So that's stage 3, stage 4. So for a patient to qualify for surgery, they have to, at least by imaging and pathology, be deemed that the primary tumor is encapsulated in the pancreas. That's the pathologist's answer. The other answer is there's been studies that have been shown that at a number of institutions, and this actually obviously isn't Jefferson, but there's a number of institutions where patients don't get offered surgery. And that's because either the oncologist or the internist or the surgeon there, so you're talking about a place like Jefferson where they do maybe 250, 300 surgeries on the pancreas a year, where there are other places not too far from here that might do 3 a year. And in those instances, you can imagine that the system just doesn't offer surgery. And they might not know that that patient should get surgery. So a lot of this, and a lot of people believe this, a lot of this is awareness. And in some ways, in some places, you hear a lot of stories that the patient goes, you can imagine a patient goes to the doctor, they have back pain, and they're older, and so the internist says, go home, did you work out? Yeah, oh yeah, I worked out 2 weeks. Oh, you must have pulled something. So they're not getting picked up, they're not getting the next group of assays and clinical assays like CA19-9 and other evaluations that would pick up that pancreatic cancer. So the reality is yes, it's based on pathology classification, but the other point as far as awareness is concerned and education is concerned, a lot of patients aren't being offered surgery.
So you have this group of sort of long-term survivors. Has anybody looked at like their HLA typing or anything like that to see if they have a common?
Yeah, so Vic, really interesting question. We find that in this case with Margaret Crook, it was probably because of a BRCA2 mutation. There's a guy, Steve Leach, who was at Memorial Sloan Kettering, who is now at Dartmouth, and then we published a paper where we actually asked that question, 3-year survivors versus 6-month survivors. So at the same time, the pathology should tell them that they're the same, but what's the difference between them? And we found some molecular markers. Your HLA question is a really interesting question because what Leach finds is that the tumor infiltrate with lymphocytes are different. And I think they are actually looking at maybe HLA as a possible reason why that's happening, or at least an immune biomarker. But that's a really interesting question. It's an intense line of research of why do we have long-term versus short-term survivors. And, honestly, other than the answer of luck at this point, we don't have the answer. I mean, it's the reality. It's a great question. Yeah.
Is the probability of getting pancreatic cancer the same across this globe, or are there populations that are much lower or much higher?
Yeah. So another interesting question. So yeah, so the main factors that increase your risk of pancreatic cancer, I believe to date, I'm not an epidemiologist, is the smoking and obesity. So smoking seems to increase people's risk significantly, and so does obesity. Interestingly enough, we found that people who were obese, though, and got pancreatic cancer, they actually did better on chemotherapy, probably based on some of the data we showed because they have higher glucose around their tumor, and so the chemo works better. But it's mostly smoking. Culturally, Ashkenazi Jewish population, because of the BRCA2 mutations, they have a higher prevalence. They're not just only predisposed more to breast cancer and ovarian cancer, but also pancreatic cancer. There's a slight increase in the African American population. We're part of a study with Hopkins trying to do the genome-wide sequencing to see if there's any correlation there in a multivariable analysis. But there's really nothing that, there's a high population in Japan, so we've kind of ruled out diet who get pancreatic cancer. So it's mostly smoking, obesity, and some subcultures like Ashkenazi Jewish, African American, but otherwise nothing really sticks out.
Other questions? Anybody else? Is HUR, yes.
Got another?
A friend of mine had 4 children, and her daughter died at a very young age of breast cancer, and her son then died a couple of years later, very young age, of pancreatic cancer. And she heard that there was a relationship.
Yeah.
Not Jewish, not Ashkenazi at all. What,
So
Would that be if it's not the BRCA?
You'd want to look at the full, what we say, family pedigree and see if I guess if either the parents do get cancer or if the grandparents got cancer. But yes, there is a relationship. So prostate, breast, ovarian, pancreatic, those genes, what we call the BRCA1, BRCA2, or the DNA damage repair genes, all have a connection to those tumors. And so they're not only predisposing patients to cancer, and they're probably carried, the mutation is inherited, but it also allows for those tumors to be susceptible to some of the drugs that I showed in the middle of the talk. But they should probably see it sounds already like they should probably see a genetic counselor, and they can all get tested. That's the best thing to do. At least meet with them and see, and typically nowadays, if there does seem to be prevalence within the family, insurance actually pays for the genetic testing.
Is HUR a tumor promoter?
Yes.
It is.
So it's yes, not just under stress conditions, but we believe, and I went through that data really fast, that it cooperates with KRAS mutations to actually cooperate and cause pancreatic cancer.
Everybody has this sequence?
Everyone has it.
So we're all born with it.
We're all born with it, and as to your earlier point, it probably allows for us to deal with daily stressors, but then the tumor cell takes that network and uses it against us.
What's the size of this protein?
It's the 36 kilodalton.
Yes.
So you had said that I believe it was the BRCA mutation had reverted back to its original wild type. Has that ever been seen before with any other type of, like the genes, where the cancer actually completely reverts back to the wild type? Or at least been observed in...
Yeah, so that was, when we published that was the first time in pancreatic cancer, but it's been shown in ovarian and breast cancer that they've specifically, those genes, BRCA1 and BRCA2, can revert back. And it actually was initially found, Alan Ashworth took cells, and one of the ways we studied resistance is he grew those cells in this therapy for weeks and weeks, and the cells that emerged, he went and sequenced and found that they reverted back. So it's pretty remarkable, because it actually shows you that the cancer genome is able to mutate like that in such a way to become resistant.
I was just saying it seemed counterintuitive to me that if you had a mutation in BRCA1, a reversion, I should say, back to its normal state,
Yeah.
and that gene was originally driving the cancer, then why are you still getting cancer?
So
Growth.
it's a great question. So my theory about that is that when you talk about the lifetime of the tumor and how the initiation happens, that mutation is probably important for that moment in time. So the analogy I tell students is it's like that event in their adolescence was probably really important, but when they're 35 years old, that event may not be as important. And so that mutation initially, it's a genome maintenance gene, probably allowed the cells to become hypermutable and discover other mutations. And by the time the tumor got what it needed, it probably didn't need that mutation anymore. And so it probably said, okay, fine, I'm going to revert back to it, and I'll be fine because my whole network's already set up. So that's the theory. But typically speaking, yes, in normal experimental settings, if you revert, so for like P53, BRCA2, if you actually correct that in a cell with CRISPR or something else, those cells will die. So it is kind of counterintuitive, but that's our hand waving answer. Yeah, and that's a great criticism. We did, we sequenced the normal, no, we sequenced the other normal, the other panel of cancer genes. We didn't find anything else, but certainly there are probably other what we call passenger mutations that happen. And so the thing that I usually say, appropriately to your point, when I present that data is, this is an N of 1. However, it's in a patient. So it's more powerful than, doing it 10 times in a petri dish. But your point's well taken. I agree.
I'm interested in early diagnosis. And so, similar to the Vogelstein model of colon cancer, you've got various stages, various mutations going on. So I guess one possibility might be, or what do you think about, since there's clearly some genetic relationship in terms of, different generations. For example, my father died from pancreatic cancer, so maybe I should be tested for some of those KRAS and other kinds of things. Not necessarily doing the entire population, but if there's a tendency, having somebody in your immediate family or whatever that's had pancreatic cancer, maybe they should, as an initial, early detection approach, look for some of these other changes. The problem with KRAS, of course, is it's mutated in a lot of different things.
Yeah.
So, it's,
So, this is a really interesting line of investigation now. And so it's called CAP study at Hopkins. Mike Goggins and Mimo Canto is running it. And what they do is they're doing exactly what you're saying is, so they're taking high-risk families, high-risk individuals who they have identified this mutation with. I mean, if it was just, I'm not saying just like it's just, but within your family pedigree, if it's just your father, you probably don't have it inherited. Usually the definition, you have to have two first-degree family members who died of pancreatic cancer to be truly defined as familial. So in your instance, if that's just it, then it's, you probably, you wouldn't get the insurance to pay for it. But for these high-risk individuals, what they've done is they've done a intense screening process. And initially, they found that the screening didn't matter, that they couldn't stay on top of it, even if they screened patients and gave them endoscopies for every 6 months. However, the recent data that Mike showed is he showed that if he can identify these patients early, we start treating them early enough, we can increase their survival. I don't think they're curing it by the standards that I mentioned at the beginning, but they're actually staying on top of it. Because, like for men, when we get PSA test, prostate-specific antigen, there's a screen there. Women, mammograms, other things. There's nothing really for the pancreas. And again, going back to the 55,000 people each year, insurance companies are not going to pay for us to get CA19-9 or MRIs or other things to get done. I will say Dr. Yo, when he gives a talk sometimes, he talks about, I'm not sure if he does this, but he talks about if he had all the money or if we had all the money and insurance had all the money, would we get an MRI every year? And he says sometimes that he does. And I'm not sure.
Yeah, it would be a good pre-screening.
Yeah. And but to that point, even with the current technology we have for imaging and early detection, it's not great. And so even when we detect there's a tumor, some people believe that those stage 1, stage 2 patients, because we're talking about, those were stage 1, stage 2 patients that only had 1% to 2% cure rate, right? So some people believe that that's not even stage 1, stage 2, that we need to move the needle back even further. And so that might have to do with our imaging and early detection. So if we can get a blood test like this WEE1 sequence or some imaging that's better, we would probably make a big difference in this disease. The Pancreatic Cancer Action Network, about 2 or 3 years ago, said they wanted to double the survival by 2020. It's 2019 now, and we moved from 6 to 7 to 8 to 9% 5-year survival. So I love them. I've gotten 2 grants from them. I support them. I love that they went for it, but it will be interesting to see how they pitch that coming 2020, because I'm not sure we're going to double the survival. But one way to do it, the reason I brought it up, one way to do it would be early detection. Yeah.
Why some patients are more responsible to gemcitabine treatment and not others? What is the reason, the role of HUR in gemcitabine?
So we initially published on that, and then as an interesting academic note, we disproved it and published against it, because what we found is in small population, we went and expanded it to a larger population, and one way to actually disprove yourself is to redo something and do it in a larger population. But that's part of being a scientist, and I tell the guys all the time, it's important for us to get it out there so no one else does. But why do certain people, so the standard of care, the standard of care for pancreatic cancer now is just chemotherapy. It's non-targeted therapy. It's throwing the kitchen sink at the tumor. So it's either gemcitabine or Abraxane, and these are just big DNA intercalators, and they just affect FOLFIRINOX, oxaliplatin, 5-FU. These are things that just generically target a number of things in a cell. If we knew why certain patients, a lot of people put forward, these are biomarkers to tell us which patients would do well. And we haven't found a good biomarker for that. And I think if we could find that out, that's another way we can increase survival time, because we waste a lot of time treating patients on therapies that don't work.
Yeah, I was wondering, is there a physiological basis why this cancer is so rapidly fatal?
So rapidly.
Fatal.
Yeah. To keep with the theme, I think it goes back to the fact that we detect it so late. And so when you compare it to other cancers, so two things. I think it's because we detect it late, and because of the theme I had out there that it's grown in such a harsh environment with the low glucose and the hypoxia that when we treat it, I think in some ways we get those cells stronger and they get more aggressive and they kill. Other tumors are they don't live in that type of environment, and so we can treat them a little bit easier. There's this tumor microenvironment. When I talk about the fact that only 30% of the tumor that you resect is actually the tumor cells, it's surrounded by these other different types of cells. So to get a drug into those tumor cells that you want is even harder as well. So there's all these lists of things that make it really difficult and fast to, I think the biggest reason is we detect it so late.
But what actually is killing the patient physiologically?
Oh, physiologically. It's the tumors growing in other settings and totally destroying, the organ. So in some instances, it affects the pancreas in the sense that it could cause someone to have diabetes, severe diabetes. It could obstruct certain things so the person becomes jaundice, and so they need a stent put in to sort of survive. But it's really these micrometastases that go off into the liver and other things that cause a dysfunction and cause cachexia that allow certain organs to just fail.
Can you picture the HUR protein as the basis for an early detection test? It seems like it would be a cheap assay, compared to DNA assays, and you wouldn't have to actually go in there and take a piece of the organ. You could probably find it in the blood.
Yeah. It's a really interesting question, because my colleague has found that HUR is in things called exosomes that are released in the blood. And so we haven't published this paper yet, but we're publishing a paper in which we show that HUR is released by cancer cells and exosomes into the blood, and the end part of the paper is potentially we can capture it and detect it as an early detection mechanism. That's very new. But it's certainly something we've thought about. And certainly things it could regulate could be things that we could detect, hopefully. And the sequence that it binds, that sequence is in someone's germline, so we could potentially detect it in someone's blood and see if they have that difference in that sequence. For instance, that WEE1 sequence. Yep.
This is sort of off the cuff, but I know there are available methods that you could, when you take a blood test and you get some of this DNA that's been cast off by the pancreatic cells. There are ways to use DNA testing to find out whether they're pancreatic cells versus other cells. So,
Yeah, I think you're talking about circulating tumor cells, so that
Um,
people can capture in their blood to see if there
well,
are.
not a whole cell, but
Okay.
I could talk to you
So
about
just DNA, circulating
Just-
DNA.
using methylation.
Yeah, you can do that, or
Methylation that's
or KRAS.
specific to
Yeah.
cancer cells.
They haven't. A lot of people are studying that, but it hasn't been proven yet. I mean, hopefully it will be. But we tried it with KRAS, and we didn't have a high sensitivity or success rate. But it'd be great if it worked. Or looking at the methylation status as well. I know people are looking at that. Yeah. Okay, sure. Always happy to talk about it.
Are there other questions for Dr. Brody? You know where to see him after this lecture. Students, follow him. Persistent, that is the question. Follow up with him. And that's the way you will be he is one of the role models for you, so you will be one of him tomorrow if you persist and follow, okay? On that note, please give him a big
Thanks
round.
for your attention. Thank